So it's different for cones. So if I just draw my cone cells like this, each one of them has its own neuron. Fantastic. That means if light lands there, then the brain knows, because this is the one that's taken off. If light lands there, then the brain knows, and it's this neuron sending the information, and the same for this one. So what we get with the cones is really high visual acuity. We're really able to see a sharpness of image. The problem with the cones is that it takes a lot of light intensity to break down the chemical that's inside them to start the action potential off. So that's why in dim light you see things in black and white, because the cones aren't being stimulated. So each type of cone cell, of which there are 3, contains a slightly different pigment, and that pigment is a type of opsin, and that opsin is literally just broken down in the same way that we have rhodopsin being broken down into retinal and opsin and then built back up again later. So there's something called the trichromatic theory of colour, and it's the idea that each of these only responds to a different wavelength of light. And you know, again, going back to your year 8 physics days, that there are 3 different wavelengths, and light travelling at that wavelength will appear as red, that travelling at that wavelength may be green, and another wavelength will be blue. So each of them are only stimulated by a different wavelength of light. So then if you've only got 3 different cones responding to 3 different wavelengths, which is 3 different colours, how do you see the great variety of colours that we have? And the answer to that is, each of them, if you're looking at an image that is red, this red one is going to be stimulated more. You're going to have more action potentials firing down there than this one, which might only be stimulated a little bit. So it's the mixture, just like when you were a child in primary school and you mix different colours of paint. If you put more red paint in there, you've got... Okay, so if we're looking at cone cells, we've said this wavelength of light responds to blue, this to green, and this to red. And we've only got 3 types, and we've said that each respond to 1 wavelength only, and it's the frequency of action potentials that determines the depth of the colour, and that is what we know as the trichromatic theory of light. Cone cells. Cone cells, each one has its own bipolar neuron, and that means that, fabulous, we're going to get a sharpness of image because we know when light lands there, it's that bit of the retina that tells the brain that's where light landed. If light lands there, it's that bit of the brain, and so on. The difference is that with the iodopsin that's in these cells, for that to break down, it takes a huge amount of light. There's no summation. So they don't operate in dim light because you need the bright light to break it down. When the iodopsin is broken down, that changes the permeability of the cell to sodium ions, and that's what's going to start off the next action potential. So, give an advantage and a disadvantage of retinal convergence in rod cells. Well, advantage, we can see in dim light. The disadvantage, we lose that sharpness of image. Give an advantage and a disadvantage of a lack of retinal convergence in cone cells. Well, we get sharpness of image, but we don't get to see colour in dim light.